Hello, everyone, and thank you for tuning in to Better Biopharma. I'm your host, Tyler Menichello, and I'm very excited to be joined today by Sianny Christanti and Dr. Babu Medi from Interius BioTherapeutics, where Sianny is the Senior Director of Process Development and Babu is the Senior Director of Drug Product Development. Sianny and Babu, thank you so much for being here.
Thank you for having us, Tyler.
Of course. For those of you who don't know, Interius is developing INT-2104, an in vivo CAR T for B-cell malignancies. And yes, you heard that correctly, in vivo CAR T. By creating CAR T cells in patients' bodies, the Interius team's mission is to cut out the complicated and lengthy manufacturing process associated with conventional CAR T and replace it with a more effective off-the-shelf option for patients. And so I'm thinking the best place to start off, Babu, for our audience, would you please describe INT-2104, what it is and how it's designed to work? And then, Sianny, I was hoping that you could talk about how that influences process development a little
Tyler, again, thank you for having us. And before I go in and describe INT-2104 itself, I want to introduce Interius BioTherapeutics. As you mentioned, we are a spin-out of UPenn, co-founded by Dr. Saur Gill, a hematologist at Penn Medicine, who treats regularly patients with autologous CAR T, ex vivo CAR T cell therapies. And Dr. Gill saw the challenges with existing CAR T therapies. And the goal of Interius is to take this directly into the patient to generate the CAR T cells in vivo in the patient. We're doing this by re-engineering the lentiviral vector platform to target T cells and NK cells specifically using our proprietary binder. Our lead candidate, as you mentioned, is INT-2104. It is based on the lentiviral technology where we have three unique features that are engineered into it. First, it is engineered to target T cells and K cells with a proprietary CD7 binder. Secondly, our platform technology includes VSVG-based fusogen, but that is detargeted against to avoid any LDL receptor binding, but retaining the fusion function after the CD7 binding happens. Finally, 2104 includes lentiviral genome encoding a human CAR-20 molecule to generate CAR-20 positive T and NK cells in vivo. And as you mentioned, the product is designed for B-cell malignancies. The product is expected to result in three big leaps, I would call it, compared to the ex vivo CAR T therapies. First thing, we do not need to take the cells out of the patient or a donor to manufacture the cells, because this completely eliminates, as you mentioned, the cell processing aspect and the associated challenges. Our product, secondly, is an off-the-shelf product. It's an engineered lentiviral vector that is stored in a regular -70, -80 freezers, just like similar to the mRNA COVID vaccine in the pharmacy. And when a patient becomes eligible for this in vivo CAR T therapy, the lentiviral vector product would be pulled out from the freezer, thawed, and prepared and administered within the same day intravenously. Simple intravenous infusion. And the third big advantage with our product is the patient doesn't need to go through any lymphodepletion chemo regimen to receive this product. We at Interius are very proud to be the first biotech that achieved first in human with this exciting new modality in the in vivo CAR T space. We started dosing patients at the end of the last year, continuing to dose escalate, and we expect preliminary data later this year from that trial.
Thank you, Babu. Sianny, do you want to add some color there about the process development for INT-2104? how its
Yeah,
properties affect that.
yeah, definitely. As Babu mentioned, the vector that we developed for the in vivo CAR T is different from the ex vivo. So one thing is we have the detargeted VSVG and also having the additional binder molecule for specificity. So how that affects the process development is definitely the plasmid has to be customized to be able to encode the binder molecule. And but beyond that, the vector itself actually fits very nicely in our manufacturing platform. So the way that we do this in Interius is, the discovery space discovery group will generate maybe multiple iterations of vectors, as they're testing it for in vitro functions, and we in the process development side get a pretty early read on these vectors, bring it, put it back in in the lab, and put it in the manufacturing our platform. And so far we see that the manufacturing platform that we've developed are very robust. Minimal process changes have been required, between the different vectors or products. In terms of formulation and storage is the same way. We basically use the same formulation. So I think this highlights sort of the we in Interius are developing the vector platform technology as well, a manufacturing platform where we can pretty much plug and play as we go along.
Yeah. Thank you, Sianny. I'm curious if you could speak to a little bit about how um, how this vector construct, I don't want to say flies in the face of conventional CAR T, but what are the biggest differences from a process and manufacturability standpoint? And then for both of you, I want to know how different this is from anything else you've ever worked on, and what you'd compare it to for
Yeah.
folks out there doing similar things.
Yeah. So I think definitely one of the main difference is obviously the lentiviral in this case is our product for in vivo CAR T, as opposed to for ex vivo CAR T is the cells, right? So that in itself is the fundamental difference. Now, what that means is, I think one for me is this basically we have to make the lentiviral in a much larger scale than the ex vivo CAR T. Um, because obviously we want to maximize the batch size and produce, maximum number of vials that we can, supply as off-the-shelf products. So scalability of a process is very important, and this goes with our strategy of, suspension process is the way to go here. Adherent process, as we all know, is you can scale them out, but you cannot scale them up. And also in terms of costs and raw material, needing serum containing medium. Those are things that we want to avoid and be able to have a robust, scalable process. Um, the other important thing is, given that the vector is administered directly into patient's body, we do have a higher bar for in vivo CAR T, vectors that we use for in vivo CAR T, in terms of impurity levels, also just a deeper understanding and characterization of the vector. And we at Interius has designed, we have designed up, in addition to designing our manufacturing process to be very scalable, we also developing, and I keep adding to our analytical toolbox in order for us to be able to better characterize the vector, as we want to make sure that the product that we're making is not only, efficacious, and but most importantly is safe. Um, the other challenge is, because we are, and this has something to do with the vector biology, and how it works in the body. Off-target transduction is a very important consideration or risk that is, potentially happening in the in vivo CAR T, but not so much in the ex vivo. So here, as Babu described, we greatly de-risk this, right, by having the binder molecule there for specificity, for CD7. And But on top of that, I think the vector biology and the vector design has to also be accompanied again with manufacturing process that could deliver that product quality consistently. So we're striving toward that, and I think we're in a very good shape there. And I would like to add that we have investigated off-target transduction in NHP, non-human primate model or NHP model, and utilizing immunohistochemistry to visualize potential, localization of target transduction. The work was actually presented at ASGCT in 2024. And the summary of it is we did not see evidence of target transduction in the non-human primate model. So that's very exciting.
This is exciting. Thank you, Sianny.
Tyler, I would like to add to what Sianny was saying in terms of looking at this differences. As Sianny mentioned, our product is the vector, lentiviral vector, re-engineered lentiviral vector. And this is very different from the use of lentiviral vector in the ex vivo space, where it is considered as a critical raw material that is still held to a higher standard than other raw materials used in that process. But in our case, our product is the final product is the lentiviral vector that is being infused intravenously in some volume, depending on the dose. So we needed to carefully think about the final product composition. That's what our group, my group does, in terms of looking at the formulation composition, and that is compatible for intravenous administration in the amounts that we're looking at, and also the formulation that can stabilize the product during the downstream processing steps, including freeze-thaws, any additional fill finish operations that we do, and also at the clinical sites, the handling and in-use stability of the product during dose preparation and administration. All of those things were considered into developing this final drug product that we're using currently.
Yeah, thank you, Babu. And I'm curious, and you were mentioning formulation and obviously delivery is a huge part of an in vivo CAR T. I'm curious maybe what the team's biggest learnings have been in re-engineering this vector to target in vivo and have high transduction. Maybe something that was unexpected or that you might not have considered beforehand before embarking down this journey.
Are you asking about any surprises with the in vivo?
Yeah, if there's any, if there are any that folks might.
Yeah, so we've obviously done multiple preclinical studies, including mice models and, as Sianny mentioned, non-human primates, where we dosed a number of NHPs in the preclinical and also GLP tox studies, where we infused the product intravenously, and we didn't see any issues related to the safety. The product has been shown pretty safe, and we were able to, again, share that NHP data where we saw the B-cell depletion. So we, again, we haven't seen any surprises in terms of us being able to dose this in vivo.
Thank you, Babu. Sianny, earlier you mentioned about analytics, and I was curious when it comes to, obviously, I'm sure your release testing is different when you're talking about an in vivo product. It's a vector versus ex vivo cells that you're testing for release. And I'm curious, or in vivo, what the differences might be between metrics and assays. Like which assays do you rely on for potency and batch consistency, and how might those differ from typical ex vivo CAR T?
Yeah, so I think from a potency standpoint, we rely on cell-based assay. Like they call it infectious titer assay. It's also probably the same type of assay that's used for ex vivo. The difference is we have to develop a specific assay for cell line that expresses CD7 for potency, because maybe the standard cell lines out there do not fit that category. So I would say that's one big difference. I have to admit that I am a relatively newcomer to the CAR T world, so I'm not particularly familiar with the ex vivo analytic panel. But I think from what I have gathered is there's definitely potency. There's a particle quantification by P24. There might be a genome quantification. There might be some limited specific panel of impurity. But I think it being a considered critical raw material, I would say that I think the specifications and the bar is definitely not as high as the in vivo. And I think you did ask this question earlier, which I never answered. You asked how this compares to our previous experience, right? So I came from many years experience at Merck, mostly in the viral vaccine side. So I would say that the in vivo CAR T, lentiviral used for in vivo CAR T, is probably closer to any biologics or parenteral drug that you put directly in the patient's body. So it definitely has impurity clearance is very important, and even if it's not a release assay, it will need to be characterized as consistency of the product, and eventually will have to be a specification for the later phases. We also characterize particle size, subvisible particles, things that is of concern for something that is injected in intravenously. Um, I think Babu am I missing anything?
Yeah.
Is there anything?
No, I would say you covered it pretty well, Sianny. On top of what is required specifically for this re-engineered vector with the CD7 binder, that's the potency assay that's different compared to the ex vivo product. But I would say for our lentiviral product, it is held to the standards of any traditional biological product that would be intravenously administered. So that's very different from when you consider ex vivo process, where lenti is used as a critical raw material. You would not be doing those kinds of testings. And as Sianny pointed out, one of the things is particle size characterization, and what is happening with the final product. How consistently are you making this product? And that's pretty important in this case. So
Thanks, Babu and Sianny. Babu, I'm curious to hear from your perspective. Sianny shared a bit about how her background helped in what she drew on for this quite new modality. Honestly, there's not too many to compare yourselves to. I'm curious how your professional career has past has influenced what you've drawn upon, what learnings you've drawn upon as it comes to developing this novel tech.
yeah, thank you for that question, Tyler. So I've come to Interius back in 2021. So it's been close to 4 years with Interius. It's an exciting time. And prior to coming to Interius, I was with a company developing allogenic ex vivo CAR T cell therapy products, Allogene Therapeutics. I was there heading the formulation drug product development for allogenic CAR T cell therapy process products. And I've seen what the cell therapy products and the challenges with those products would be, and that's certainly eye-opening in terms of these new kinds of products, how it can benefit the patients, and the accessibility and the manufacturability and the ability to supply what is needed. And before that, as Sianny said, I was also at Merck working in vaccine development space. Specifically, I worked on viral vector based vaccines. So that certainly helps us look through in terms of in vivo lentiviral vector products. That's certainly very helpful to have that experience and product knowledge. Feel.
Yeah, that's great. Thank you. Um, Babu, or really both of you, I'm curious, a lot of the conversation around CAR T and especially in vivo CAR T, it centers around biology and delivery, but I'm curious to what degree manufacturability is considered. Do you see that as a key differentiator in the space? Um,
Yeah, Tyler, I can take that. I would say yes. Apart from developing a safe and efficacious product, manufacturability is very critical for consistent quality product and reliable supply at the scales you need for these products. I would say manufacturability in terms of product consistency from batch to batch um, is certainly better in terms of in vivo products such as ours. Using lentiviral re-engineered lentiviral vector products compared to cells that are being manufactured outside the body, and there is a lot of challenges with that, meaning there's donor to donor differences in the process variability that comes with it. That can play a big role in terms of the quality of the cells that you're getting out of the process. So, and in terms of allogenic cell therapy products, where you can, or the goal is to generate several doses from each manufacturing batch, which again, I think the goal there is to make multiple doses. I don't know how far we can go in terms of getting those doses high, but it is still challenging there. There's issues in terms of scaling it up to a level that we need. Whereas in our case, the scale up with the viral vector or lentiviral vector manufacturing process, we think we can make hundreds of doses per batch. Of course, it depends on the dose required eventually, and also the cost of goods can be significantly improved with the scaled up process, while maintaining or meeting the critical quality attributes as we scale up. So on top of it, the product stability and supply chain is also very critical. In our case, the cold chain requirements is less stringent, if you will, as opposed to the ex vivo or any cell products that would require liquid nitrogen conditions and storage conditions, shipping conditions. Whereas in our case, we store our product in a regular -70, -80 freezers, just similar to, like I said, other vaccines. There are some vaccines that are stored that way. So that also plays into the manufacturability, and so I would say, yeah, as our CEO Phil would say, manufacturability is as critical as being able to develop a safe and efficacious product.
Thank you, Babu. Sianny, I wanted to hear your thoughts on manufacturing a little bit, but more so in this, and you mentioned before how the company's transitioning to a suspension cell line. When when I had when I interviewed Phil and Tim last year, we talked about how the company initially decided to go with an adherent cell culture platform to expedite clinical delivery or clinical entry, but now they're transitioning to a 2,000-liter suspension process. That was last year. I'm curious where the company stands today and what have been the biggest hurdles in building up that suspension process, whether it's cell line adaptation, media optimization, or whatever the case may be.
Yeah, I think everything, that you mentioned, cell line development, media optimization, bioreactor selection, parameters, all that are additional things that we have to tackle, obviously moving from adherent to suspension. And I think we have we have been blessed with a very strong PD team that we have built. Some of my my team members they have experience in previously with CHO suspension for monoclonal antibodies, and some will have some experience in live virus. So we definitely use this collective expertise to kind of guide us through on the developments of the suspension. We do focus on sort of the high impact activities. What I mean is, like, we being a small company, we cannot do media optimization the way that maybe a larger organization with a big infrastructure could do. So, in this case, we will choose to use more of a commercial media and do some optimization, in terms of feeding and trying to kind of augment the performance of the media. In terms of the cell line, we took the approach of adapting a cell line in-house, and this is done to avoid sort of the financial obligation that comes with royalties. And we have been successful at that in developing the suspension cell line, and have made a master cell bank that we will use moving forward. So I would say maybe, and that was a lot that, there was just a few things that we had to tackle. But I would say probably one of the biggest challenge that we have has to do with a large scale transfection, where we will we had to investigate then optimize the way that we add the plasmid to the cells, and how do we do that, what kind of mixing we will need, what pH, what conductivity, and so on. So I think it's those are actually probably one of the most challenging issues that we have to solve, especially as we go larger in scale, and where the volume will be larger, and obviously the time that it will take will be more variable as well, and on the manufacturing floor.
Yeah, thank you, Sianny. Follow-up question to that. I was curious if there were any lessons that from adherent process that are translating well, or not at all, as it comes to the building the suspension platform.
Yeah, I think as you said earlier, Phil, Phil had said told you that we took the approach of using adherent production platform to kind of get us quickly in phase 1. And we had to develop a process for that. Now, the strategy there is we actually designed the process in such a way where it includes, I would say, redundancies in the step, including orthogonal methods of removing impurities. So, for example, we might put in 2 chromatography steps. So that is the basis of our suspension development. And actually, what we found out was we actually didn't have to change the what we call adherent process too much, in the downstream side, which shows that basically the process that we developed earlier was quite robust. There are different impurities that we had to deal with between adherent and suspension. So, for example, adherent, you have bovine serum albumin that will come from the FBS source. In suspension, obviously, we don't have that. But at the same time, from a suspension culture where we have, more than 10 times the number of cells, we will have a higher load of host cell proteins. And all of that combined, actually, we have been quite, I guess, pleasantly surprised to say that the process actually can handle, the quite drastic variation of the process. We do have to tweak things a little bit, but we don't have to, do a major overhaul of the process, which is really good. So that goes to show that the manufacturing platform that we develop is quite robust.
Yeah, I was going to add quickly, Tyler, that on top of the downstream considerations, obviously, when you're changing the cell process, we did look into the final product, drug product, and stability of the final product as well, along with those changes. So again, the formulation is robust that it can handle, or we can show that there's no differences in the product stability, and um, there's no need to change the composition.
Yeah, thank you. Thank you both. Um, I want to keep talking about manufacturing, but I also wanted to take a step back and ask a regulatory question here for those in the audience who may be more interested in that. I'm curious, for whoever wants to answer this, what regulatory considerations are unique to an in vivo CAR T therapy like INT-2104? And how is that shaping CMC and comparability planning?
Yeah, Tyler, I'll start off, and then Sianny, you can chime in as well. So, as this is a brand new field in vivo cell therapy, but I would say regulatory considerations are a big part as we are charting a new course here with these novel, first-in-class kind of products. Um, One of the challenges with any new modality in first-in-class products is that you're in a way pioneering this in a few different ways. While you're really looking at and focusing on proving the biology or testing the new biology and understanding the mechanism and safety of the product, we're also sort of paving the way in terms of regulatory path, because we're both agencies and us are learning together and adapting. One big difference with, again, in vivo CAR T therapy is that we're infusing the engineered lentiviral vector directly into the patient. So whereas the vector used for ex vivo is outside the patient, so it has to be characterized fully, as Sianny touched on previously, for safety, purity, potency. I would say as a traditional gene therapy or biologic product, right? So apart from the design of the vector to specifically target the T cells, we needed to carefully consider the residuals from the process and their levels. We need to ensure that the final product composition is compatible. So we, again, assembled a CMC team that is well versed to develop the process, and that can meet those requirements. The drug product team, again, we spent time in terms of looking at the excipients and that is required to stabilize the product. So I think, overall, I would say, in terms of dealing with the development of a first-in-class product and regulatory interactions, so far the regulatory interactions and the feedback that we got is obviously very critical, but it's also very positive and constructive. As you can see, that we got approval to dose patients both in Australia and Europe at this point. So that requires a lot of regulatory interaction and consultation during this whole time. So I would say, yeah, there are some guidances that you can take into consideration, the existing guidances from regulatory bodies. And there's also new things that we're dealing with. So that has to be really brought up early on and consulted with whatever regulatory agency that you're dealing with.
Thanks, Babu. Sianny, anything to add to that?
No, I think like Babu said, we kind of take into account maybe different regulatory guidances out there, whether it be vaccines, biologics, and also on the cell therapy side. I would say that we maybe are more on the side of the um, biologic side where you administer that directly to patient. But even that, we're not just taking that guidelines and directly applying it to the in vivo. I think there's some considerations with the risk and benefit. For example, like if you think about vaccine, they're administered for healthy individuals and at times for infants and and children. So those guidance not necessarily apply directly for our application lentiviral for oncology space. So I think there's definitely like Babu said, that we take these guidances very seriously, but we also have conversations with regulatory agencies to kind of have a more of a conversation, of what is acceptable, phase appropriate, what is appropriate for phase one, where do we want to go toward for later phases. So it's not just we got it perfect the first time. We kind of build in, sort of relationship and kind of build data set and kind of ultimately, hopefully, in the commercial stage, we will have a more defined sort of specifications for everything. So it has been a journey, I would say, the regulatory side, like Babu said, because we are embarking on these, novel approaches. I think this is kind of goes with the game. So it's been a lot of learning for me personally. So it's very very eye opening and exciting. So
Tyler, Tyler, to touch on your other question about comparability, how this fits in. We're obviously, as you heard from Sianny, we're moving from adherent to suspension process, and there are certain other changes that happens as we develop these products. And, we, as discussed earlier, we have analytical toolbox that we use to compare. And as we change any of the process steps or anything else, so we also are, obviously interacting with the regulatory agencies, with the seeking scientific advice with the any proposed changes. And again, so far our interactions has been pretty constructive and positive.
Yeah, that's great. Thank you, Babu. I'm curious, you just alluded again to the development of the suspension process. So I was curious about scale there. I was curious at what stage you anticipate being able to reach full scale GMP production for INT 2104. Sianny, what scale do you think will be sufficient to support your early clinical pipeline? Interius is pipeline.
yeah, I think our goal is to get to the full scale by phase two. We're pivotal studies, right? So we want to minimize scale change between phase two, three to commercial. And I think this is a best practices that is, you might have heard of this, the process is the product. So we minimize changes as much as possible at the later stages. And understandably, in the earlier stages, things are a bit on the flux. And I guess this applies to us as well. Right now, we're in the middle of phase one, where the study is designed as a two-part study for dose escalation and then later as a dose confirmation. So we are running at a smaller scale than the what we think will be the full scale, and we will run the as the required number of batches depending on the dose that's required to support the phase one.
Great, thank you. I know we're running short on time here, but I wanted to ask two more questions. The first being, when you're talking about regulatory considerations, Babu, you were talking about how you're you're pulling a bit from gene therapy. I guess we have the benefit of it's kind of it's straddling between a cell therapy and a gene therapy, and it's quite unique in that way. So I was curious if there are any promising trends or enabling technologies that Interius is keeping its eyes on um, from the broader field of gene delivery and gene therapies. Like, what advancements there have maybe found their way into your product or process design?
Yeah.
Thing you're keeping an eye on, and love to hear your.
Yeah, no, that's a really good question. Obviously, we're currently very much focused on getting our clinical data readout from our lead product candidate, INT-2104. And as we learn more about the safety and function of this product, just like any other product development, we are also keeping our eyes open and looking at other ideas that are moving into the clinic recently, and how they would compare and what is needed to get the job done. So we are obviously keeping our eyes open while we're focused on getting our own initial preliminary clinical readout with our product. This is a brand new field, in vivo cell therapy, as we call it. Although the products are different, it could be viral vector, it could be different flavors of the vectors, and there's also non-viral approaches that are being looked into for this, I guess, in vivo field. We obviously continue to, hopefully we will obviously learn from each other as a field as this data flows in. And also internally, we have been and we continue to work on researching new concepts and new ideas of improving this in terms of either it could be the safety or the efficacy of the product. So that and to bring that might result in potentially new drug candidates, right? So again, our goal here is clear that we want to develop products that can be delivered faster to the patients, meaning off the shelf, and better, no need to lymphodeplete, and generate better CAR T cells within the patient, and cheaper with the bringing down the cost of goods and scaling up the manufacturing. So again, we're going to learn from each other as the field evolves, but I feel very fortunate to be working with a great team here at Interius that is striving hard to make this a reality one day.
Great, Babu. Sianny, anything to add there?
No, Babu sums it up pretty well. I am grateful for the opportunity to be in this field. I feel like I learned so much. And like I said earlier, I'm a relatively newcomer, I would say, to the cell and gene therapy. So it's been learning for me, but I'm also excited that I could contribute to it, as my perspective from having been in the viral vaccine and biologics side of things where I could apply some of these knowledges. So personally for me, it's been rewarding, but I am excited as our team and as I guess as a whole, as our biotech landscape here, where we are all trying to come up with new therapeutics ultimately for the patient. And I think it takes many organizations working on similar things and kind of learning from each other, like Babu said. So it's been an exciting journey and kind of watching what's out there, what's going on. So but yeah, we are I think this year is a very important year for Interius as we are undergoing the clinical study. So we're excited to see where that takes us.
Thank you, guys. Well, I want to ask you our closing question here, and it's something I ask every guest, and I want each of you to answer it. But from where you sit, how do you think we can better biopharma? You could take any angle you want.
As with any new field, obviously you cannot get it done alone. You need to learn from each other in different concepts and ideas that are coming into fruition, meaning getting into the clinical testing and development. And I also feel like we're small biotechs. We're grateful for this investors and supporters that have believed in us and in terms of getting us to where we are. I would say, obviously, as we move along, we would certainly need to bring forces together to take this into late stage and make it commercial reality and make it available to the patients as soon as possible. So it takes a lot of effort. We're a small team here, but we are a strong team with the right skill sets to get the products designed and tested and moved into the clinic. I look at it within 3 years time frame. That's really fast getting into the clinic with a new first-in-class modality. I think it takes a lot of ideas and innovative ways to get that done. So there's a lot to learn from each other, and obviously regulatory agencies, they also play a critical role in this. And so far, we've been fortunate to have that constructive, positive feedback on what we're trying to do here.
Thank you, Babu. Sianny, any closing thoughts on how we could improve the industry as a whole?
Yeah, Babu sums it up really well. I think for me, maybe what strikes me here, just as you asked this question, is just how we should consider everything as not like a fixed goal line in terms of, so say the regulatory, right? We have to think about what is phase appropriate. At times, I think people strive for perfection, while excellence is maybe sufficient for certain. It's a fit for purpose. So I think just having an open discussion about it, kind of make people think and maybe challenge some of the status quo, or just thinking about different approaches. I think as we always will come up with new approaches, and there are some things that are stake in the ground that we cannot move, right? But there are some things that maybe in the past was was in place for a specific instances or specific scenario that might not apply to you. So I think it's just having an open mind and the courage to ask questions, and and just seek to understand is very important. I think that will make us all better at doing our job, and more efficient, and getting there without wasting maybe unnecessary resources or unnecessary churn. So yeah, that's the only thing that comes to mind at this moment, Tyler. So
Well, you guys are definitely challenging the status quo.
yeah.
That was great. Sianny, Babu, thank you so much for joining me here on Better Biopharma. We really appreciate it.
Thank you for having us.
Our pleasure. Thank you.
That was Sianny Christanti and Dr. Babu Medi from Interius BioTherapeutics. As always, I'm your host, Tyler Menichello. Thank you so much for joining me today on this episode of Better Biopharma, an extension of Bioprocess Online. We hope to see you next time. Thank you.